This is a multiple sequence alignment. This is the identification of residue correspondence. In sequence alignment, it is the identification of residue correspondence in sequences. Any assignment of correspondences that preserve the order of residues. That is, if it is a string, that order should be maintained of residues within the sequence is an alignment, and in an alignment gaps can be introduced. Sometimes there will be some missing areas, so the program will introduce a gap for that. Then first type of sequencing is pairwise sequencing, what we did yesterday. Then we know two terms, one is homology and the other is similarity. Homology means it is an inference or a conclusion about a common ancestral relationship, that is homology, whereas similarity is the percentage of aligned residues that are similar in physicochemical properties such as size, charge and hydrophobicity. This similarity, the influence of size, charge and hydrophobicity, it comes when we align amino acid sequences. When we align protein sequences, it will consist of amino acids. Some amino acids may have same property, so these factors comes into consideration. Then homology, it is a qualitative statement, whereas similarity is a quantitative statement. Then similarity versus sequence identity. In sequence similarity and sequence, though they feel like synonyms, but they are synonyms for nucleotides, but they are different for protein sequences. As I said earlier, similarity, it refers to the percentage of aligned residues. Then the methods of pairwise alignment can be global alignment and local alignment. So global alignment, this diagram shows a global alignment in which the whole sequence is considered. In both the cases, if we have two sequences, sequence 1 and sequence 2, both the sequences, the whole sequence is considered while doing the alignment. So two sequences to be aligned are assumed to be generally similar over their entire length. The entire length is considered while aligning. Then the alignment is carried out from beginning to the end. It is from beginning to the end. Then it is more applicable for aligning two closely related sequences of roughly same length. We have to consider the length of the sequences which are to be aligned. Then this method may not be able to generate optimal results for divergent sequences and sequences of variable length. So here it fails to recognize highly similar local regions. That is one of the drawback of pairwise global sequence alignment. Global alignment. Then in local alignment, it does not assume the two sequences by their length. Here the length is not a factor. So only finds the local region of similarity. Here the sequence similarity is considered, see, in the similar regions are considered here. Then it is more appropriate for aligning divergent biological sequences. Like even if they are of different species, diverging species, we can compare them by local alignment. So here what is considered is the local similarity of the sequences, not the whole length similarity. Length is not considered in local alignment. In global alignment, the whole length of the sequence is considered. Of the sequences, both the sequences. We are comparing two sequences. Then local similarity is considered in local alignment. Length is not considered in local alignment. But in global alignment, the whole similarity is considered and length is a factor. So these are the types of algorithms that can be used. They are dot matrix plot. We can see sometimes similarity searched as dots. They are indicated as dots. Then dynamic programming method and word method. These are just the theory part of it. You don't have to deeply go into it. This is what is a dot plot method. Similarity searched in as rows and columns, just like matrices. See, this is a dot plot similarity search method of two sequences. I think you don't have to go deep into this. These are all dot plots, and these are some of the inferences in dot plot method. If you have a straight line joining the diagonals, then it is a perfect match. Then if you have repeats, then there will be lines, there will be a central line and lines outside them, like parallel lines. Then a palindromic sequence will be a cross mark. The other is partial palindrome. Then microsatellite will be like this. Then mini satellites, many parallel lines. Then homologous sequences, they will have gaps in between. Then indel means some part is similar but some part is, maybe some gaps are there. Then disadvantages of dot plot. I don't think you have to go into this. Some programs, I have shown this because some programs use dot plot method for sequence identification. Like emboss package. Maybe when you read some papers, you will come across these terms. Dot matcher, matrix plot, all this. These, they depend on dot plot method. Then next is dynamic programming. This is the most used method. Dynamic programming and word method are the most used method. Here the sequence alignment is done with amino acids. For dynamic programming is mostly used for amino acids. And here the score is used. Scoring method is used in dynamic programming. All these are theory classes, so I am just running on it. Then dynamic programming for global alignment. You may sometimes come across terms like Needleman-Wunsch algorithm, Smith-Waterman algorithm, and so on. All these are used by different softwares or different programs, and they use dynamic programming. See, this GAP, G-A-P, it's a web server. It is a web-based, pairwise, global alignment program. It uses dynamic programming. These are some other web sources like SIM. It is a web-based program for pairwise alignment using Smith-Waterman algorithm. Then S-Search, it is also a dynamic programming method based on Smith-Waterman algorithm. Then LALine, it is also an alignment program which uses Smith-Waterman algorithm. But unlike S-Search, this LALine gives specified number of best score alignments. You can use it, it is available. The differences between global and local alignment, one is Needleman-Wunsch algorithm. Both are for dynamic program, using dynamic programming. but one is global alignment and the other is uses local alignment. So I think this is what is sequence alignment. Now we can see what is BLAST. BLAST program. Whenever we write BLAST program, we will write a reference. What is it? Whenever we write in a manuscript, BLAST program, sequence similarity was searched using BLAST, then you will have to write a reference. What is it? Ashwell et al. It was developed by Stephen Ashwell of NCBI in 1990. It is available in NCBI. Then its objective is to find high-scoring ungapped segments among related sequences. So here the similarity is searched using pairwise alignment. Even though there are different sequences, a large number of sequence similarity, they use pairwise alignment and each sequence is searched pairwise. Then these are the basic steps. I think we can go through the system also. We can simultaneously do with the system. I think you have the BLAST sequence which we downloaded yesterday. FASTA format which we downloaded yesterday. Okay, we can use that for, this is step 1 is seeding. There are different steps. It is written in a sequential manner. It's for understanding and sake of learning. So here what we have, they are saying to create a list of words from query sequence. What we have is a query sequence. These are the different steps. I think I don't have to deeply go into this. We can do the exercise. Here in BLAST they use the word method, not the dot plot or dynamic programming. They use the word method. Word by word is searched. Sequence is used as words. And then they will give a score, HSP, that is the high scoring segment pair, or it is called the maximum scoring pairs. I will leave my slides here. You can access it whenever necessary. Okay, these are the variants. Let me see. Did you take the NCBI website? There you can see BLAST. There are variants of BLAST. We are doing only nucleotide blast. You have downloaded a nucleotide sequence, so we are doing nucleotide blast. So you can click nucleotide blast. You may have, many of you have done it. How many of you have done blast? Almost all. Okay. So what we have to do is either you can copy paste the sequence here in this blank column, or you can upload a FASTA file. Choose file and you can upload from here. Or if you want to blast an already submitted sequence, then you can type the accession number. Also, one more thing, you can create your user ID and password, or you can log in using Google Gmail ID, our Gmail ID. You can log in to NCBI using your Gmail ID. So your searches and your history, search history will be remaining in the website. Okay, so you can do that also, so that your data will not be lost, because you can see it from your search history. You can retrieve it. Otherwise, it may not store all the blast result or anything. So we are going to do blast N, and I have chosen a sequence. Then here we are doing only I am doing only 16s. If you are doing something else, you can change the collection. See, here you can change the collection, but here I am using nucleotide from the nucleotide collection. If you have something else, then you can, if it is RNA sequence, then you can select it as RNA, reference RNA sequence, representative genomes, reference sequence, genome database, whole genome sequence, which is whole genome data, then you can do whole genome, like that. Okay. Then if you have any specified organism, it is optional. You can enter your organism. Then if you want to exclude some cultures like uncultured or environmental samples, if you don't want to see any sequences submitted from metagenomic data, then you can remove those data. Like sometimes the similarity will be more with uncultured, and if you want to omit that from blast analysis, because sometimes when you do the analysis, all the sequences will be similar to some uncultured sample. Then you may not get the original one, so you can avoid that also. Then sequences from which type of, then entrance query, if you have any entrance query, like I have shown you yesterday the query terms, search query terms innale kaanichanu. Yes, in my yesterday's class, how to retrieve data using entrance. Query terms undengil you can add it here. Then you can optimize the search. Highly similar, more dissimilar sequences, then somewhat similar sequences. We are doing mega blast, we are doing highly similar sequences. So you can click blast. You can also optimize the algorithm parameters here, like how many sequences are to be targeted, then expected threshold value, word size, what is the limit of the word size which is to be selected, like that. But we won't usually do anything here. You can also add filters. If you suspect that your DNA is also amplified, or you we have to suspect that also, because when we are doing without gloves or something, some sample may have mismatched, may have been added by accidental contaminations are possible. So we can omit that also. You can add filters, then just blast it. So this is my job title. My job title was NR1. It was an already submitted sequence. You will get the result here. It is showing similarity to E. coli strain NF4, 16S ribosomal RNA gene. This is a partial sequence, and it is showing 100% similarity to E. coli itself. Then query coverage is 100%. That is the length which is covered by the query in the original sequence. Then E value is the error value. Then percentage identity is 100%. Then length was, here the length was 1348, but see the others are whole genomes. You can see the length is more. Okay. Then next you can see the graphic summary. Graphic summary, here you will get 100% coverage, means the graphic summary, it gives the length, or it gives an idea of the coverage. If the length which is covered or the alignment score is less, the color code will vary. If you are submitting or trying to find out a new sequence, new novel one, it is not been deposited too much, then it will show, it won't be showing high similarity. Here we are doing E. coli, so it is available, and if the data is not so much available and the alignment score is less, if you are doing a fish barcoding, then the database may not be populated. So sometimes the similarity score will be less. Then the color code will vary. These are the color codes according to the alignment scores. If it is very less, we usually don't consider below 80 to 200. Below this green, if the graphical summary is in green, we won't consider. We will go for another sequencing. Okay. This maybe there are some errors, or the organism may be different. Some difference will be there. So we won't consider that. Usually in fish barcodes we get that pink color, 80 to 200. Then we will get the alignments here. Those sequences which we have seen and the submitted sequence. These are the alignments, and here you will get the taxonomy of the organism. Also you can download those alignments. Alignment session. Alignment is, I will show you. See, here the E. coli is aligned to NF4. The accession number is MT649781. So our query sequence is aligned to this NF4, that sequence, and the alignment is shown here. Okay, like that each alignment is available. If it is compared to 100 sequences, 100 alignments will be available. See, this is the subject and our query sequence and the subject, and down you can see each sequences like that, each alignment like that. What? Expect value. Usually it is error value, E value is error value which is considered.
And this too, the percentage identity is 100% or nearly 100%. How do we apply?
You mean the query coverage is not 100%, okay.
But the percentage identity is 100 or maybe 99.
Okay, nearly similar.
Yes.
Then we can say that the query coverage, it depends on the length of the sequence which is compared.
Suppose it was around 800 bases.
Maybe our sequence was of shorter length and other sequences, you mean like that?
Okay.
So it's okay, the identity is what is important, percentage identity is what is important, but we can only we can come to a conclusion only when the query coverage is also high. Yes, we have to take into account of that, because
is there a minimum threshold that this much query cover has to be similar?
No, the query coverage is what our query sequence is that much available in the database. That is what is compared. So when our query sequence is less, even then it will be 100% in the database. So the query coverage will be high if our sequence is not pure, or some impurities, or the sequence, maybe sequencing errors are there, then we may not get this 100%. That is the problem. So if we have errors in sequencing, we have to check. Either we can do filters, we can add filters, because sometimes maybe the sequence may not be pure, then there will be errors. That is one of the reasons of less query coverage.
But the major reason is that
Then
lack of
your sequence may, if your sequence is novel, then also there will be less query coverage. If you are identifying a new organism, then also there will be less query coverage. So if you are sure that your organism is a different one, or the gene which you have amplified is a different one, then you can go with your assumptions, hypothesis, and like that. No issues.
How can we know the sequence? How can we check the sequence?
We can add filters. If you have any difference while adding filters, then we can conclude that it's having impurities. And in ABI itself, usually they will give us pure sequence, like they will trim the sequence and we will get only pure. But sometimes what the sequencing service agencies, what they say, claim is, if our culture is not pure, if the culture is having some impurity, then there may be differences. That's what they claim.
Usually the beginning and the ends of the sequences are a bit not clean.
Yeah. So they will trim the data and we will get two data. One is row sequence and the other is trimmed sequence. You don't know? Usually we get two types of sequences. One is row sequence and the other is trimmed sequence. When we know how to play with it, like how to trim a sequence, then we can manually trim the sequence and make it pure by using ABI file and like that.
Sequencing agencies are really not doing it. They are just giving things.
No, we are getting trimmed sequence. They will give us trimmed sequence. If you are not confident with their trimming, then we can go for ourselves. Like we can do ourselves.
With other organisms, so how will I choose among these searches? Which one will I choose? It is closely related to that.
You have to go for more sequence identity than query coverage. So if the sequence identity is more, you have to download it, then go for multiple sequence alignment and then phylogenetic tree, so that you can see from which lineage it comes from.
The percentage identity is similar to organisms in the blood, or will we choose among that?
Percentage identities similar. Yes, sometimes I'll come to Bacillus itself. If Bacillus sometimes series will be similar, it will show similarity to series, then sometimes series and along with that some other species also. Then you have to choose all those organisms, download their sequences, and then you have to construct a phylogenetic tree, and you have to find from the phylogenetic tree where it comes from, which is the ancestor, same lineage, and which is near to the organism of your query. Then you have to come to a conclusion. It is the species level identification. Number of matches in alignment is the number of sequence matches. This means here everything is matched. This is in 16S. It is showing similarity to 16S, but if it is showing searching in a whole genome, then some other regions will also be matched. So you can see in the other one it is 7 matches. So the match number of matches will be high. We usually look for this blast result as such, not the alignment. We will just check if it is aligned properly, and this type of confusions will arise only when the sequence is not similar. If there are dissimilar, like when we are submitting fish barcode sequence, like that. Not for 16S. 16S is not that much confusing, but error prone. That is the only issue. So you have to just go through it to see if the alignment is proper. If there are so many gaps like that. I think it's clear. So we can see what are the variants of BLAST. In BLAST programs, BLAST N is the query of nucleotides. Then BLAST P is the use of protein sequences as queries against protein sequences. Protein sequence is queried against protein sequences. Then BLAST X, it uses nucleotide sequence as queries and translates them. Then BLAST X, it uses query is nucleotide sequence. It is searched against protein sequence, translated and then queried against protein sequences. Then T-BLAST N. T-BLAST N is again queries are protein sequences into a nucleotide sequence. Then T-BLAST X is, that is the translation of BLAST X. Okay, translation of BLAST X. That is, it uses nucleotide sequence which are translated in 6 plane reading frames to search against nucleotide sequence. These are to identify proteins. If you have a nucleotide sequence, it is queried against or to identify proteins. So these are the variants of BLAST. It is available in the same website. And one more thing is that you can have the phylogenetic tree from BLAST itself. You can select the sequences and then you will get a phylogenetic tree from BLAST itself, but it is not that accurate. Genome. These are whole complete genome sequences. Yeah, it's differences there. These are complete genome sequences. It's written also. So here you can unselect, untick all those and select those sequences which are of your interest. Like 1 E. coli and nothing else is there. So the sequence on you can go with an E. coli, Shigella. But whenever you go for tree plotting, you have to consider one thing, that the sequences should be of almost similar length. The sequences which you are trying to export for phylogenetic tree should be of similar length, almost similar length. Also please take care that it is not of whole genome sequence. Then you can go for one uncultured bacterial clone, uncultured bacterioplankton, I think enough. Then you can go for a distance tree. See, because these are all same organism, it is not having a common ancestor or see, Shigella, it is not a rooted tree or anything like that. If you select some different organisms and then go for a tree, you will get a proper phylogenetic tree. It is only showing the distance. Tree method is minimum evolution. You can select neighbor joining, FASTA algorithm, which can also be used for sequence alignment. FASTA alignment, which can also be used for sequence alignment, but we usually use BLAST for sequence alignment. I don't know if it has bioedit, so you can do it from bioedit. So for that you need a you don't have any forward or reverse sequences, right? Full length DNA sequence, usually we will prepare full length DNA sequence from our forward sequenced data and reverse sequenced data, and then it is used for BLAST analysis. So this is the icon of bioedit. You can just open it and try it. From the file you will get 2 sequences. You have to save the sequences in .fas to open. You may have saved the sequences in .fas. Earlier I have told you while we were doing pairwise alignment, this .fas, and then you have to open the forward or reverse, whichever, both the sequences. Then you will get, it will be opened in another window like this. One will be forward and other will be reverse. Two different sequences. They are not the forward and reverse. If you yourself have any forward and reverse sequence, you can use it. This is the forward sequence. Then you can open the reverse sequence also. This is the reverse sequence. So it will open in another window. Then what you have to do is, there is a mode in BioEdit. Did you open BioEdit? So when you open the sequence, there is a mode which shows select or slide. You have to select the sequence like what we did while doing pairwise sequence alignment. Make it, change the mode to edit. Change the mode to edit mode. So first you have opened the forward sequence, and then you are opening the reverse sequence, and you are selecting the reverse sequence, and then you are copying the reverse complement from the edit button. You copy the reverse complement. You copy the reverse complement. Have you seen reverse complement in the edit? Okay. Then it was already copied, and then in the other file you have, in the forward file, we have opened the forward file. So right click it and paste the sequence. So the complementary sequence will be pasted. The complementary sequence will be pasted. It came in the window itself. Sometimes it works odd. BioEdit, sometimes it works odd. Okay, did you paste it? Then you will get both the sequences in the same window. We have to copy this region, and whenever we do some copy paste or something, we have to change the mode to edit mode, and the other tab to overwrite. Edit and overwrite. Pointer tab. Mode is edit and the other one is overwrite. Then you can copy the sequence from the missing sequences and paste into the gaps. See, if there are gaps, you have to fill it from the other sequence.
Where we have to paste?
You have to paste in the gap of the forward sequence. The first one is, if the first one is forward and second one is reverse, you have to fill the gap from reverse to, that is the reverse complement to forward. Yes, select it by just using mouse. You can select it, or you can use the shift arrow key while selecting. Just right click, copy, and then select the other region and then paste it like this. Select this and then paste. Remove all the gaps in the sequence. Copy. It's not working. Maybe some systems, in some systems, the full options of BioEdit may not work. I don't know the reason exactly, but in some cases it won't work. The full length sequence without gaps is your sequence, and then save it as FASTA format. You can save it again as .fas file and use it for submission. You have to select the sequence and then select the other gaps and paste it over that. Select this, copy it, select the gaps and then paste over it. Understand? Fill up those gaps. So while filling up those gaps, you have to change the mode to edit mode and overwrite, because we are overwriting the gaps. There are different options. What else are there? Overwrite and other 3. Usually we used Sequin for sequence submission, SEQUIN. It is also freely available. It was freely available from NCBI, but it's no longer used. Full length sequence kittiya, you can delete the other sequence and you can save the full length sequence as .fas again. Save it as full length, name it as full length sequence.
Name only, not the whole sequence. You have to select whole sequence for the name only.
For deleting. Just select the name and then you can delete. Right click and then you can delete. You have the full length, the other sequence is your full length sequence. You can save it as .fas. From file, here in the whole window you will see the file. Save as .fas and you will get the full length sequence. Okay. You can open it in Notepad also, like I said yesterday. Okay. So you can use this for blasting. Full length sequence is often used for blast identification, blast analysis, and also for phylogenetic tree. Then next is sequence submission. Also for submission also we use full length sequence. Now the submission is based on another tool, Bangit. Sequin is no longer used for sequence submission. NCBI advises Bangit for sequence submission. So there are 2 tools. One is through a submission portal where we submit ribosomal RNA, rRNA, ITS sequence. So both these we can do the submission online. Now it's working online. We don't have to do so many sequence corrections and all. We can do all the things online, which is the organism that we are doing. All those things or all the details we can enter online and use the online portal for submission. And if you have any queries, you can email them. The email ID is given in the NCBI website itself. You can email them. They are highly responding. They are very supportive and you can submit your sequence. Okay. I think my session is over and you will have next sessions afterwards. Okay, thank you. If you have any queries, you can either email me. My email ID is bindiya79@yahoo.co.in. Then my mobile is 8547449388. Okay, WhatsApp is also available in that number. Okay, thank you so much.
Good afternoon, everyone. And now we are moving on to the second session of the day. And I'm extremely privileged to introduce our resource person of the session, Mr. Avinash, to the gathering. He is currently working as Senior Research Fellow of an ICMR project at the Department of Biotechnology and Microbiology, Khalkut University, Kerala. He has completed his PhD thesis entitled In Silico Assessment of Inflammatory Hormone Antagonists at Khalkut University. He also works as an assistant professor in biochemistry at NHS College of Arts and Science, Vadodara, and as a trainee in Center of Bioinformatics, Kerala University. He has published several research articles in peer-reviewed journals, so I wholeheartedly welcome you, sir, to handle the session. Please, sir.
Good morning to all. Let's do a small experiment to model a protein based on homology modeling. Okay? All of you heard about the homology modeling? Did you heard about homology modeling? Anyone? It's based on the basic principle that we model a protein with a non-structure. We have a protein sequence that does not have a protein structure that can be predicted by homology modeling. In this session, we do in the online platform. First, we want one protein sequence. So, you are familiar with the NCBI website. Okay, then you just search for one gene, any of gene. Please do it. Any gene you are interested, like protease, something. I want a gene sequence in all of your computer. Ayurveda is basically treated on tridoshas, Vata, Pitta, Kapha. In modern medicine, that changes it to targeted based, molecular target based drug designing. So, we need data like protein structures and chemical structures. So, that is important in this drug designing process. Nowadays, this drug is mainly based on single compounds, sometimes a mixture of different compounds. Presently, nanomedicines are getting market. Coming to history, first of for the world in the world, the drug is based on mainly plants or natural product. The one most important example is foxglove that is used to treat the cognitive heart failure. And coming to the history, one important discovery is that Alexander Fleming discovered the penicillin from the mold Penicillium. I use these like target. Target means molecular recognition site to which that drug binds. Target may be in protein or DNA. In this workshop, I prefer the target based on protein. Then the protein may be different types like receptor, enzymes, transport molecule, ion channel, tubulin, and immunophilin. The drug discovery classified into four types. First one is ligand-based drug designing, or otherwise is named as indirect drug designing. Then next one is structural-based drug design or direct drug designing, and third one is rational drug design, and last computer-assisted drug design. Structural-based drug design, that is commonly used to drug designing strategy. It is helped to produce new drugs. It gives the information about structural dynamics and electronic properties about the ligands from calculations like algorithms and scoring functions. Mainly, here the structural based drug designing is work on the lock and key principle. You know the lock and key principle, like the specific key is used to unlock the lock. That's why that is the main principle behind the structural based drug design. Here, two types of structural based drug designing are the one is ligand based drug designing and other is receptor based drug design. Receptor based drug designing means building ligands based upon the receptor's binding site. Ligand molecules are built up with different constraints based on the binding pocket by assembling it into small pieces in a stepwise manner. These pieces can be either individual atoms or molecular fragments. The main advantage of this method is producing novel structures or novel drug. Here the next term, docking. What is mean by docking is try to attempt to find the best matching between two molecules. Like earlier I mentioned that the finding right key to the specific lock. Here we need two biological molecules. One is the target receptor and other is specific binding small molecule. They can interact each other. Here the docking process identify the orientation of ligand on the specific binding site. The flowchart of the steps involved in structural based drug designing. Initially, we screen a number of ligand molecules from database or our own designed molecules. And the second step is the finding of a protein structure. It is mainly based on we mainly use three techniques for the prediction of the protein structure, 3D structure. One is X-ray crystallography, another is NMR, and third one is homology model. It is purely based on computational. So, after the prediction of 3D structure, we go for the prediction of active site with otherwise the pockets where the ligands going to bind. After that, we analyze the interaction based on the scoring process. The higher scoring process we select as, and further experiment is carried for the drug development process. After that drug development process include preclinical trials like animal model studies. Then if that particular drug compound have minimal side effect or acceptable ADME properties, that is selected for the further clinical trials. Then that is shifted to the market. This one is X-ray crystallography. Like I already mentioned that. Here, the homology modeling, that is also known as comparative modeling. Constructing an atomic resolution of model from the target and an experimental three-dimensional structure of a related homologous protein. Different steps included in the computer-aided drug discovery. First one is target identification, then target validation, lead identification. Lead means a drug or small molecule. Then lead optimization, then preclinical trial. This is a representation of finding a drug in different strategy. First one, the first stage is include drug discovery. The second stage is preclinical development, and third one clinical development. Then regulatory approval. Then go to the market. It includes 4 steps. First one is protein preparation. Second one ligand preparation. Third one grid box generation. for this docking. In protein preparation, we need the protein structure, 3D structure of protein that we can get from RCSB database. You are familiar with the RCSB database, PDB database? I will show. Then ligand preparation, the chemical compound we will get from chemical database like PubChem, Sync, likewise. And we do the docking in two strategy. First one is simple docking. We in that technique we use one protein targeted against one small molecule, and the other one is the virtual screening process. Virtual screening process, we need one target protein and number of n number of ligand compounds. I think our system, I don't know what is the strength of the system. If the system is fast, we can run 100 compounds targeted against single protein. If it is not available, we can cut short into 10 compounds targeted against one protein. Then please download any gene, any protease, caspase, any gene. After completing that step, please inform me. Oh, here this 43 amino acids, below 100 amino acids, like albumin. Or any viable protein like COVID-19 NSP30 or NSP40. COVID-19 protease. Is the purpose to run the homology model. Okay, so if you have any unknown protein sequence, we can run as a case study. If you have any protein sequence in your mind, then we can explore that structure.
Sir, ഇവിടെ 100 ഒക്കെ വരുന്നുള്ളൂ.
In another tab, please open Swiss model. Swiss model. Swiss model. SWISS hyphen model expert.
പിന്നെന്താണ് ചെയ്തത്?
On that page, you can find the box. Paste your protein sequence on that box. Place your protein sequence on. Then go for search templates.
Yeah, it's a long.
So all of you have the structure have some darker regions represented in green. You can scroll down dash percentage like 96.52. You can find. So above 50% represent good structure. You build a good protein structure. If your structure have anomalies, that can be seen in the unfavorable region, that is the outside the darker region. So you must redo the protein structure model. I think I found all of your screen, all of them are fine structure, so you can use it for further studies.
You know the structure of the protein amino acid. So in that amino acid we have an amino terminal end, we have the carboxy terminal end, we have the central carbon atom. So you can see phi and psi values there. Okay? So the phi value is the angle between N and C alpha, the central carbon atom, and psi value is the angle between the C alpha and C. So in this particular quadrant that you see is you have represented the phi and psi values between minus 180 degree and plus 180 degree. So any molecule will be technically possible only if that angle is persistent with the amino acids. So in case of glycine and protein, I hope you know the structure of glycine and protein, which has a functional group H for glycine. So it is conformationally, it has maximum range. That is why glycine is included in there. And proline is conformationally restricted. That is why that part is, that is why specifically it is being given there. About what we have seen is, there are specific ranges for these alpha helices and this thing. The position possible. So when we are building models and templates like this, so sir, just like how sir has mentioned, so we need to fit in molecules and we need to model the structure. So this should technically work. Then only the structure will be possible. And if the structure is possible only, you can define a function for that later. So it is necessary that these angles are specified. Okay, that is the biochemistry part of it. So, and protein levels of protein structure, we have the primary, secondary, tertiary and quaternary structure. So a molecule will be possible only when it exists in its native conformation. So that native conformation is only possible when these angles have specified values. And according to this Ramachandran plot, they should always, if that molecule should be possible, it should be between minus 180 and plus 180. So that is what is structurally assisted. Thank you, sir.
Initially we selected a gene. That is the sequence I told you to download the gene sequence. Okay, I told you because in some diseased condition, our gene sometimes our gene get aberrated. Based on that gene aberration, we get just in some case some proteins are overexpressed. So our initial information is the protein sequence. From that sequence, we build a protein 3D protein model. From the theory, we know that the protein is active in the 3D structure. All the proteins are active in the 3D structure. So using this 3D structure, we can analyze what is the best molecule to activate or inhibit that particular protein. This current section we are going to do the docking process by using Autodock. Initially we need a protein 3D structure. So that purpose, please open the RCSB website in your web browser. The same the cursor on the screen, or you just search in your Google with PDB database. Please go with the search SARS-CoV-2 main protease. You can find a number of protein structure. SARS-CoV-2 main protease in complex with telaprevir. It is an antiviral drug. All of them find this page. Any problem with your computer? 6ZRT. Then go for download files in that PDB format. Select the PDB format. First one. Download files, and second one PDB format below the FASTA sequence. And another database, PubChem. It is a library of chemical structure. Here you can find a number of chemical structure. Please use this keyword aspartame. And go for download. Here, download. Go for download. Then 3D conformer. Please make a folder in your desktop. Right click, new folder, and rename as A, and paste your protein structure and compound structure on that folder. Copy your folder path. Right click on your folder A, then copy address. Then go to Autodock tools, click to file, again click to preferences, Then click to set. We are going to the directory path. Go to file, click on preference, set. On that window, there is a startup directory. Yes. Just paste that address. Yes. Then set startup directory. The right side set. You can find your protein in your desktop. Then use your mouse scroll. You can zoom it. Okay. In the protein, first of all, we should prepare our protein, because during the crystallography process there is a lot of water molecules present in the protein structure to solve for the solvation purpose. So we should delete all that additional water molecule, because when we go for docking, we can get unwanted interaction with these water molecules. So to prevent that interaction, we should initially delete that additional water molecule from the crystal structure. So first step in the protein preparation is that delete the water molecule. That can find in edit option. Please click on edit. In that delete water, 1, 2, 3, 4, fourth option, delete water. Then next step, adding polar hydrogens. Go to hydrogens, then click on add. You can find a small dialog box. From the click on polar only. Okay, then you can get additional hydrogen in your protein structure. These blue and white shades are the additional protein structure. Hydrogen. Then next step is adding charges. Here we select Coleman charges. In this software, these Coleman charges specifically used to charge the protein structure only. Then the compute gas trigger used for charge the ligand molecule. So we select this option, add Coleman charges. You can get a dialog box like, here I got total Coleman charges added 4.773. Click OK. Then again go to charges, check total on residues. This is to check whether the charges distributed equally. Here I found these two residues not spread the charges. So I click spread charge deficit over all atoms in residues. Then click dismiss. To ensure this charge distribution, again do the similar step. Yes, the protein is perfectly charged. No residues with no integral charges found. Then go to grid. Select macromolecule. Then select choose. You can find your structure name in the dialog box. Path, file, read molecule. Already done this. Then after that, open your protein structure 6ZRT. Then first go for delete waters. Delete water. Done. And next go for hydrogen. Add. Then polar only. Okay. Then go for charges. Add Coleman charges. Then click on grid. Click to macromolecule. Then choose. Then you can find a dialog box. Click on 6ZRT. Select molecule. Then you can get a dialog box like initializing ZRT.pdb contains non-bonded atoms. Click on OK. Then rename that structure with protein.pdbqt. Choose your file on desktop A. Then click on the save as type. Save as type. And click to PDB. Shall I proceed? Wait. First you change the type PDB. Okay. Then click the folder A. Then type 1.pdb. Okay. Then click save. Then close the software. Open the Autodoc tool. Open the Autodoc tool. Click on ligand. Input. Open file to PDB. Select the one named file, then open. In this Autodoc tool, here all the ligand preparation were done in single step. After choosing this ligand option, you can read this message like added Gastier charge. I already mentioned that Gastier charge for ligand preparation, and after this it merged 26 non-polar hydrogen and found in 12 aromatic carbon. Here the ligand consists of 15 rotatable bonds and torsion cutoff set to 15. Then click OK. Then click on ligand again. Then click to output, save as PDBQT and rename as ligand .pdbqt. Please ensure that you use this keyword protein preparation. Now we completed the ligand preparation. Now I think we wind up this session for today. Tomorrow we will do the grid generation and docking plus virtual screening.
And hereby we have came to an end of today's session, and rest of the session will be continued tomorrow. And thank you, sir, for this informative and interactive session.